I definitely do a similar thing. And people are like, I don't want to learn anymore. I'm like, no, you always need to learn about the ocean.
Hold on, I have to share that fun fact. Oh, there's a sea pig in the bottom left corner. That's a type of sea cucumber.
Yes, cute
And,
little sea pigs with tiny tube feet. Actually, they're not that tiny.
Massive tube feet.
Massive tube feet.
They're called hypertrophied because they've gotten so big and they can be used as walking appendages.
That is impressive. Because tube feet are pretty tiny and sort of controlled by, I want to say, hydraulics.
Yeah, that's right. So yeah, tube feet are powered by water pressure. And so all echinoderms have a water vascular system, which helps circulate oxygen throughout parts of their body, but also can be pressurized so that they can inflate or kind of decrease the pressure in those tube feet and therefore straighten them or bend them. And tube feet can be modified in different ways. So oftentimes we picture them like we would see on a sea star with little suction cup-like ends, or just sort of flattened surface area. But I know of a shallow subtidal sea star that has spiky or pointed tube feet, and that's related to how it needs to be able to move through the sediment.
Oh.
And then the sea pig has massive tube feet.
Giant tube feet. Did we look up where the name sea pig came from?
Dan, are we waiting for the boat? Are we waiting for the boat?
What group are our sea pigs in?
What group? Well, I did know that.
What was the question?
What group are sea pigs in? It's like some elasmopodid, I think, or elasmopodid. Hold on. They're the genus Scotoplanes.
Okay.
Oh, Elpidiidae and Elasipodida. Order Elasipodida.
And who are they most related to, if we were thinking of things that we had seen?
Well, they are an echinoderm.
They are
So they're related to
echinoderms.
starfish, or sorry, sea stars,
Sea stars.
urchins, but they're part of the holothurian group, the sea cucumbers.
Okay.
We have a sea spider on the center lower, I think.
Oh, yeah. Yep, Pycnogonid, just going off screen.
That's us. That's us. That's Argus.
Are you guys telling jokes without us?
It's about to cross the line.
They're looking at the sonar.
No. How about now?
How about now?
Yeah, now I can.
Oh, it's gaining on you.
Ah, okay. So what Frank is talking about, and Joe is cluing me into, is...
Also known as Miles and Wayne.
Miles and Wayne, yeah, who have decided to change their name
They're
for
alter
this
egos.
apparently shift. Yeah, alter egos.
Get a little punchy at 4 in the morning.
Yeah, goofballs. We're looking at our radar, and so what we're seeing is actually Argus on our radar.
Hmm?
I don't know if you guys in the
Sonar,
back can
yeah.
see that. Sonar.
Yeah.
Yeah.
I'm sorry. I'm also not awake yet.
It was also where they wanted to be. They stopped the boat because that's where we wanted to be.
Oh. But we were already there.
Who's we?
The royal we.
We. So we had a question about the small, fast-moving, erratic, dark objects that appear occasionally moving across the bottom. I think those are those little red jellies, but they don't appear red till they get super close to our light because that red does not attenuate all the way through the ocean. It's one of the first colors that we lose, that light, that wavelength of light. Oh, there we go. So we've got it up on our quad feed, and you can take a look at what we're giggling at. At about 6 o'clock as it goes by, that is us looking at ourselves. That is Argus. It's not the Kraken, you guys. It's Argus. There's a lot of things going on here.
You're
Would
doing
someone
0.3
else like to describe this image? Frank, you over there?
What's the question?
Tell us more about what we're looking at here at the sonar.
Frank, I've put sonar up on the set feed 3.
So that is, well, there's a bunch going on that screen. So there's our 2 Mesotech scanning sonars, one on each vehicle. And below that is a little winch tension readout that we developed this year. But Dan keeps reversing scan to look at how Argus is closing in on his position so he can go faster.
Gotcha. Dan's ready to go.
So far, 20 minutes, we started moving the boat at shift change, and Argus is finally coming along. 3,000 meters away. 3,312 meters away.
There's a countdown.
So does that mean if we see something and want to stop, it'll take 20 minutes?
That's a roger.
Or longer.
Oh.
Yeah, times that by pi and add 20%, you might be close.
Now, why is that?
Well, currently we are 120 meters behind the ship. So if we stop, Argus and Herc have to cover that 120 meters. Then we get to look at whatever we wanted to look at by probably backing up to where we saw it. And then we have to start up again, and the ship will be, 100 to 150 meters away before we actually start moving at proper speed again. So that's, it's like an hour and a half evolution to do that.
So the latency is due
Argus
to
is
the depth of the cable?
35 meters behind Herc. So 150 meter layback.
Closing in.
Yeah.
Can't hear you.
Yes, you can think of the ship. The ship moves at the surface, which is really far away from here, over 3,000 meters away, and you have a cable with Argus down below that. But it's not right below the ship. If the ship moves and Argus was stationary, it would take a while for that movement to translate down to have tension on that on the tether to pull Argus along. And so you can think of it almost like a pendulum. If we were to stop, Argus would keep moving. If we were to stop the ship, Argus would keep moving. If we stop the ROV, Argus will keep moving. And it'll take time for everything to kind of settle and equilibrate in the direction that we're trying to make it go.
Argus wants to hang right below the ship, but as we move along, sort of the angle changes just a little bit. in shallow water, you know, that angle increases really quickly and Argus moves fairly quickly with the ship. But at these deep depths, you know, we are 120 meters behind the ship, and the angle of the wires maybe only changed a degree or two.
So that is a different kind of grenadier. I think that might be Corphonoides leptolepis, the ghostly grenadier. Maybe because of its coloring. It's white in the front and sort of mottled in the back.
Yeah.
We see a lot of different grenadier in the deep sea. And at this site, we see about 2 to 3 species.
Let's see, there was the abyssal grenadier.
Yes.
I remember that one.
Corphonoides armatus, and that is sometimes hard to differentiate between Corphonoides yakine, which is the rough abyssal grenadier. Not too creative in the two different names.
Ooh.
You have an angle of the dangle app?
I imagine that app would need to know the diameter of the cable. I guess a lot of it is the surface area and resistance.
Huh?
Yeah, so we're interested in these slump sort of outcrop features to see if other life is on them, like corals and sponges and maybe an octopus or two. So we're going to explore these this morning for the next 4 hours, and then some.
And then some. Yeah, so we're in the thick or in the middle of a 30-hour dive, approximately. So we've been moving along the southeastern skirt of Davidson Seamount, around 3,200 meters to 3,300 meters. It was last year that we started doing this, and that's near the very end of our dive, we stumbled upon the octopus garden, which was a dramatic change of seafloor surface and also the kinds of life that we saw in those areas. So we're continuing along to see what other kinds of features we might see along this southeast skirt. And right here we see one of these, a bit of clay that's been exposed. Likely it's clay all below a thin layer of sediment here. And the clay looks like, oh, maybe not. That might be a rock. I was going to say the clay might be what corals are able to attach to. That's a black coral, Bathypathes. And then we have a sea anemone, Bolocera, there as well. That's the pink. And Bolocera has these really, really long tentacles, which I'm going to guess is to help them with capturing food. That's what tentacles are used for with sea anemones. You also see little brown tubes strewn about in the sediment. Those are fecal, those are feces of sea cucumbers. Let's see, what else do we see? Oh, there's another phylum. So we have echinoderms nestled in a crevice to the left of that Bathypathes coral. So those are phylum Echinodermata, and they're sea stars. Echinodermata translates to spiny skin, and that's because echinoderms have an internal skeleton with ossicles made of calcium carbonate. Hello, Bolocera. I have an idea of which way the currents are flowing based on those tentacles. Looks like there's also some tube worms that are just at the top of that small rock. You can see the fan of their sort of feeding and respiratory structures sticking out.
All right, thanks for the zoom.
Coming wide. Full wide.
All right, we have got a video question. Are you ready? Joe slash Wayne?
Sure.
I noticed that the ROVs from other ships do their white balancing in blue water, but others do it on the floor. Any particular reason for the difference?
It's, well, we do it on the floor. I was under the impression that when you white balance, you want to white balance as close to the environment that you're going to be using the camera in. So it really wouldn't make sense to me, for anyone to white balance in an environment that was different than what they would be actually shooting in. So unless there's something different or special about their equipment that once you white balance in blue water, it calibrates itself for the change in the environment once they get to the bottom. It's a possibility. Some of the cameras are pretty sophisticated now. But the purpose of white balancing is to teach the camera what white is in the environmental conditions that are obviously changing, dependent on temperature, depth, and
Can we get a zoom on that as you
Sure.
fly by? Ah, so those are mushroom corals. Heteropolypus is the genus. And that's a colony. So each of those fingers that you see sticking out from the sort of cap of the mushroom are different feeding individuals called autozoids. And then the cap itself, the sort of rough pinkness in it, is a bunch of smaller individuals that are reproductive. Thank you for the zoom.
So it is like colonial in nature, or they're just sort of two different?
They're all genetically identical, but they have individual polyps. So yes, a colony. And those polyps can be differentiated to have different functions or different forms. So the stalk of the mushroom coral is also an individual, but it's not feeding and it doesn't reproduce. Its function is to be structure, to raise up the autozoids, feeding zoids. And so it has to have nutrition provided to it by the feeding zoids.
What a team.
There's a nice feature here. Seeing a bunch of little things.
Yeah, this is interesting.
It's a pretty stark difference from when it's just open sediment on the seafloor. You get the littlest bit of hard substrate and suddenly you have all kinds of animals attaching to it.
As we keep exploring, remember our lasers are 10 centimeters apart, so that gives us a good way to measure, as well as gives us a better perspective of how big or how small these creatures are.
Oh, there's another one of those spiral corals.
Oh, it is.
We saw that
Wish I
at
knew
the end
what
of
that
our
was.
last shift.
Octopus.
And an octopus.
All
Hi,
right.
octopus.
Where is he? Oh,
Missed
He's up in the
you.
top. Missed you.
Missed you. Oh, yeah, we haven't seen one in a few hours now.
Can we zoom to see if it's a male or a female? Or do we not have time?
I'm ready to zoom. Yeah. How about you, Dan?
Sure.
Okay.
What is the...
Female.
That's good there, Wayne. I'm
In my
following.
non-octopus expert eyes, I think this looks like Muse octopus again. It's got the 2 rows of suckers, and I
Female.
don't see a hectocautilus.
I do not either. I would agree with that. So we have a
So
female.
it's a female.
And she's
Alright.
hanging out in that weird sort of stick your arms up into the world pose that we think maybe she's sort of sniffing or feeling for the environment around
Okay.
her.
Maybe there are others around. Let's zoom out, see if we got others.
Oh, we can go see.
Okay, coming wide.
Moving on.
Otherwise, you're on the wrong feature.
Oh, y'all are testing my...
She's on her way. She's
My Spanish.
Slow
Party's
going.
just over there.
The party. We gotta find it.
Ooh.
Oh, there's a bigger feature.
Ooh. What have we got here?
Also, we don't need to zoom, but I just want to point out at the bottom of the screen, you guys can see the skeleton of a coral. So sometimes when the coral dies, the hard skeleton can remain cemented to the substrate that it was on, and it'll kind of stay as a, well, a hard surface. So it can get
Pushing it.
colonized by other animals. Oh, that is a nice looking sponge. Okay, happy birthday to me. That's what I was hoping to see.
Yeah, there you go.
It actually looks like 2 different sponges, one down below and then the one up above.
all right.
Thank you.
Yeah. Oh, he's
Fulfilled
coming
my
in.
birthday wish. Oh, it's another sponge to the left of it as well.
Oh.
Covered up in sediment, but that's a sponge.
So is that still a living sponge, or is that one sort of providing structure for the other things around it?
It's hard to say. Parts of it are alive. I can see white tissue inside of it. You can see also the sponge on both sponges are covered in brittle stars. So sponges are animals themselves, but they're also really good structures or surfaces for other animals to live on and in. I think those might be benthic ctenophores at the
On
top
top. Yeah.
of that white sponge.
All right, thanks for the zoom.
Okay, coming wide.
That's a stalked crinoid up there, that yellow guy. So crinoids are also related to sea stars and sea cucumbers, so they're echinoderms, and they are specialized for suspension feeding. So they have long arms that have little processes sticking off of them called pinnules, and those pinnules help them to increase their surface areas so that they can have more water pass by more parts of their body and they can capture food. And then their tube feet are modified to help move food that they've captured down toward the mouth. So unlike a sea star, the mouth points upward in a crinoid.
I received a question in Spanish that was asking us where we were today. And so I'm going to do my best. Estamos explorando el monte submarino del Davidson in California
Spanish.
Central. So rough accent, but we are currently exploring Davidson Seamount in
Predator
Central
didn't get-
California,
Yeah.
off the coast of Central California, in El Mar.
It's a very neat step feature here, and you can see all kinds of different animals just in this view. So down in the bottom left is a predatory tunicate. So it's a sea squirt, which are mostly filter feeders, but that one is a carnivore, has this big mouth, opens up. And we could see 2 different kinds of sponges in view. One a large tube-shaped sponge, and then another one that's up in the top center of the screen, and then another white one down in the bottom right. Looks like we have another one of those spiraling corals as well. We're not sure who that is, but it's a coral. And we see another Bolosera sea anemone as well.
I was just informed that the crinoid is the Missouri state fossil.
Really?
So that's pretty awesome.
I like that a state fossil would be
May
an
have to
invertebrate.
boogie.
Yeah. Go ahead.
Missouri, I wonder, I don't know the geological history
We
if
could
they had
step the
an
boat a little
inland
closer,
sea.
maybe.
It's not going to happen quickly.
A little floaty.
250 meters, 300 meters ahead of you?
Yeah. You're 40 meters away from the, we're 40 meters away from the cliff, so I'll just come down. Easier to move Herc than I don't know how long this feature goes on. Looks like it curves in.
Yeah,
So
I'm
maybe
about
we're-
to stop and then start moving to the east.
Really?
When you guys get a chance, can you cycle power to the oxygen DO temperature probe?
Yeah, copy.
Want me to do it? Power cycled.
Thank you.
When you guys are saying power cycled, is that sort of turning on a circuit to have those probes have power, or something else?
It's turning off and then back on.
Gotcha. Sort of resetting.
General troubleshooting.
Gotcha. Oh, look at those anemones.
There are quite a few of them there, both
Beautiful.
on the clay and also on that coral.
There's a few different species there, yeah?
Likely, yeah.
Oh, look at that. At this depth, what is the average amount of notable organisms per square kilometer or mile? I don't know. I'm not counting. Taylor Ann, are you counting that?
Not to that extent,
You're
but
not? Aw, well,
that's someone's job to do after they watch the transect video
Yeah,
over
they-
again and count specific organisms. But I do keep a count if I notice there's like an unusually large amount in like maybe one screen grab or I try to count all I can, but per square mile or down to that type of thing, no, I can't
yeah, that's
keep track.
That's a lot for us to do. That will be something that they definitely kind of look at later on. But while we've got a live stream going, it's hard for us to go back and double check and make sure and count everything.
Well,
So
would that just be macrofauna, or do we
I
want to know
think
myofauna, microfauna?
There's lots of
this
different
request
things.
is macrofauna.
Okay.
Or actually, I'm gonna.
Yeah, because we
It's
could.
we would designate them megafauna, creatures greater than 5 centimeters.
Oh,
That's megafauna?
Generally,
Wow.
I mean, people do vary on what they define it, how they define these different things. But
Huh.
yeah, macro. Oh, hello, Saccocalyx. There's a sponge.
oh, look at this.
So, yeah, megafauna are critters that are large enough to see and handle, about 5 centimeters or larger, things that you can see from ROV video. Macrofauna are animals from 5 centimeters or whatever sort of larger cutoff you have, down to 300 micrometers, so 1/3 of a centimeter. And so those are things that, you know, we would find if we were to take a sample and then sift it through a sieve and see what's retained on a 300 micrometer mesh on that sieve.
Oh,
And then below macrofauna is myofauna, which I believe are down to about 50 microns. Pretty sure. I don't. And then you can even go below that. So I don't know that we would call
I think
anything
there's
nanofauna. By the time you get
there's a
that small, you're
tulip
looking at
sponge.
microbes.
Or-
Oh, you're right. There's a tulip-shaped sponge at the bottom left of the screen.
Yeah, it was the
Very
first
cool.
one.
And then-
It's the first one I've seen today.
Yeah,
A lot of anemones here.
quite a few.
Yeah, look. This feature is very cool looking. Sort of a very stark cliff on our Davidson Seamount.
A cliff is great habitat for suspension feeders to live on. It would be hard for a mobile animal to move around on it. They can climb, catch prey, but really living on a cliff gives the advantage for suspension feeders of having a lot of water flowing by, so they have access to more food. And wow, this is tall.
Yeah.
This is very
It's about 15 meters. It's going to pop down for a look.
Okay.
Come
Great.
back up.
wow.
Great, that's great.
What? I can see the bottom, right?
The cliffs of Davidson.
Wow, the shelf.
All right.
There's a bunch of small sponges there on the left. Sea anemone in the middle, and a little red shrimp.
Huh.
Is that a snailfish?
I think it's a snailfish.
Ooh.
It's that little guy.
Wow, it's pretty neat. You have the most animals right here at the ridge of this feature.
Uh-huh.
Some impressive holding on power. Sort of defying gravity.
Sorry, I lagged that. Sorry.
The
I'll come back
anemones
towards you.
seem to be
Wow.
happy at that one line.
Mm-hmm. Crinoids right there sticking out.
Looks like a
On the-
giant eyelid.
Yeah.
This is a very neat feature. So this rock that we're looking at is all sort of volcanic, right?
I'm not sure that this is a rock we're looking at.
Yeah, this might
Okay.
be clay or sediment slump.
Oh.
Yeah, so it was the little hints of the clay that we were seeing before as small ridges, and here it is much more exposed.
Okay.
I
So
don't know
if we were to poke this, would it have a softer texture-ish?
I would assume so.
Oh.
It would definitely be softer than rock. It might still be compacted clay. So it would be like if you squeeze clay from your yard. It can be hard once you squeeze it really tightly, but it'll still squelch through your fingers a bit.
That's really cool. I would have just guessed that that was hard.
Ghostly grenadier?
Ghostly grenadier. Corphinoides leptolepis.
Oh my gosh.
You did it.
I'm learning my grenadiers. Except I have no idea what the rough abyssal grenadier looks like yet.
Oh, someone said, I'm pretty sure the anemones are trying to spell out happy birthday, Amanda.
Oh,
Turn off your auto
how
heading light.
thoughtful.
So,
What nice anemones.
Yeah, they're very sweet.
Pull it on you. Can't get to where I want to go, so.
They do look like colorful bows on a gift of an ROV dive.
Just for you.
Am I sharing a birthday with any other viewers out there?
Oh,
October birthdays?
good question. I don't know. Viewers, let us know if it's your birthday. Happy birthday.
It's a former third officer's birthday today.
Really?
Mm-hmm.
Aw.
I definitely got all of my days mixed up and missed one of my friend's birthdays by a day. Fortunately, he forgave me, but he had a birthday 2 days ago.
It's tough to remember when you're on a ship even what day it is, so I'm
Yes.
sure he was understanding.
Yes, I definitely thought it was the other day and didn't realize we had already been on the ship for so long. I was like, oh, my bad. He really wanted to sneak into my luggage and come on the boat with me.
Well, hopefully he's been able to tune in and watch so he can at least participate this way.
Yes, I think so. At this depth, are there limitations to the size of an organism?
I couldn't think of any hard and fast limitations.
No.
Yeah, in general, there's even a pattern of miniaturization and deep sea gigantism that can occur in deep water. So like the sea spiders or pycnogonids that we've seen, they can actually be much larger in the deep sea than what we would find in shallow water.
Now, is that to be able to get more food or sort of travel further around for more food? What do we think is the reason for such giganticism down here?
I don't know the answer to that. I wonder if there are hypotheses about that. So,
I
one
don't know
deep
the
sea
answer
animal.
to that either.
Yeah. One deep sea animal that's really well known for being gigantic is the giant deep sea isopod.
Ugh.
So let me
Yes.
see if there are hypotheses about its gigantism.
Gotcha. Isopods are super cool. They do look like little hovercraft when they fly around, but there is something... Ooh, they give me the heebie-jeebies.
Oh, of course, why didn't I think of this right away? As I'm looking up what hypotheses are out there, there's a researcher, Craig McLean, who spends a lot of his research time studying deep sea organisms and the relationship of body size, or the evolution of body size, and sort of what can drive that. I just pulled up a paper where he's the lead author.
Ooh, that sounds interesting. I actually just finished reading a book called Size Matters, and it was an idea of looking at sort of evolution, biological change based off of size rather than other features. And it was really interesting because there were components that were required to change in order to have animals of such large sizes or such small sizes.
That is interesting.
Yeah. I forget the author. Let me look up the author.
So the paper that I pulled up, here, Craig McLean is the lead author. This is in the Journal of Biogeography, published in 2006. It's called The Island Rule and the Evolution of Body Size in the Deep Sea. And in it, they tested the generality of what's called the island rule, which they refer to as a graded trend from gigantism in small-bodied species to dwarfism in large-bodied species, and whether that island rule applies in the deep sea. So islands also can have gigantism or dwarfism, miniaturization that occurs. And they hypothesize that it's based on selection on body size in an environment where there are limited resources, and that the body size of deep sea species or of island species converges on whatever optimal size or whatever size is optimal for their particular ecological niche or requirements in that habitat.
What's an example of miniaturism?
Oh.
Well, I think it's all related to how large, relative to how large they are in shallow water. So in this one, they talked about gastropod or snail species that are large in shallow water that are absent in deeper water. So Busicon is one gastropod, Bula is another, that has shallow and deep water species or shallow and deep water range. And so looking at that range, they saw that there was miniaturization that occurred for those gastropod species.
Very cool. And if you want to read more on that, you can look at the book Why Size Matters, From Bacteria to Blue Whales, and it is by John Tyler Bonner. And that was this very interesting perspective on size in our biological kingdoms.
That was a fun question to think about.
Yeah.
I really like all these questions coming in. I mean, I usually don't know the answers, and then I get to go and do a little foray into the literature to see what's been done or what's known about it.
Yeah, giving us some puzzles this morning.
Mm-hmm.
Well, we have another one that we kind of discussed yesterday, but I thought it was a very interesting discussion. So do animals have a similar day/night cycle? So we have a bunch of animals that are down here in the dark all the time. So you wouldn't necessarily think they would have sort of day-night cycles, but we were discussing yesterday the idea of diurnal migration, where they would cycle up to or migrate up to out of the depths during nighttime, so they could feed and not be seen by predators. And then these creatures would move back down during the day, so they would have some sort of sense of a day-night cycle.
Yeah, and some of the fish travel 1,000 or more meters every day up and down the water column during that diurnal vertical migration.
That must be why they abbreviate it DVM. That's a mouthful.
Yes.
Actually, I was really
Yeah, go ahead, Dan.
I was really interested in that question after it was asked yesterday, because I've been looking at patterns of behaviors of animals that are stuck to the seafloor. So sessile animals like cnidarians, sea anemones, or zoanthids, and sponges. And it turns out they have patterns of behaviors or movement as well, or activity levels. Anyway, I stumbled upon this paper where they looked at trawl data from trawls near the bottom and trawls collected at the seafloor and in the Mediterranean Sea. And they looked at what time of day or night those trawls occurred. And it turns out that there was a difference in what was caught in those trawls, even on the seafloor, from day and night. Yeah. And they hypothesized that was related to animals on the seafloor being able to burrow and therefore be out of the way of a trawl. And then coming out when either their predators were gone from DVM or some other related kind of event. So that was cool. There's actually a lot of circadian cycling to them. Oh, and our circadian rhythms are driven by melatonin levels in our bodies, and so you'll have, well, okay, shouldn't say driven by, it's driven by light, but our bodies release melatonin to kind of signal or indicate the changes that need to happen throughout the day to have a circadian rhythm. And another paper I looked at found melatonin levels changed in 2 deep sea fishes across the day. So, yeah, I don't
Do you
know
remember
how.
what those were?
No, but I want to say it was grenadier. Let's see if I can find that paper again.
Oh, that's kind of cool.
More sea anemones lining the edge of that clay
Let's
ridge.
see them. We got another big coral there.
Oh, there's another predatory tunicate. Those guys are so neat. I would love to study those. I want to know what's changed in them, like in their feeding structures. So most tunicates, they're filter feeders, and so they bring water in through one siphon, and they pass that water over what's called their branchial basket, which is essentially gills with a mucus mesh across them. And so bits of food are caught against that mucus mesh, and then the mucus is rolled up and moved by cilia toward an endostyle, which is gives a path to the mouth, and so they can eat their food that way. But for the predatory tunicate, it still has a branchial basket, but it doesn't need to use it for capturing food because instead it captures larger prey. I want to know if they still have the ability to catch small prey or if they just use their gills for respiration or what. I think that'd be really neat. Ah, and I found the paper, Erica. So it was
It was Corphinoides.
Corphinoides armatus and then also
Synaphobranchus.
yes, I'm glad you're looking at these names.
Yeah.
Which it refers to as the deep sea eel. Do we have those here?
We might have something like Synaphobranchus in California waters. I don't know if it's a Davidson.
Is it a true eel or another one of those cusk eels that are actually fish?
I think I'll look it up.
Okay. Look at this. Everybody's hanging out on the side of the sea cliff. So you guys said this was sort of a thicker clay substance. How would this cliff form then? Would it just get really sort of heavy and avalanche the side off to have this sheer edge?
That's my best guess.
Okay.
There are slumps that can happen, and yeah, avalanche-like flows that can happen in the deep ocean.
So Synaphobranchidae is a true eel, and it is found at Davidson Seamount.
Whoa!
We don't see it that often. It's elusive, so we would like to see it again.
What makes an eel? What is an eel by definition? Is that just a particular group of fish?
It is. It's an
Sure.
order of fishes, and I will let you know.
Okay.
Yeah, there's a few unique things about eels. I'm pretty sure that it's got sort of one continuous dorsal fin. Yeah, we better check the details before I start giving you false information. But I do know that some eels have sort of a second jaw inside of their mouth. And so they can grab food with their normal jaw and with their teeth, and then they can move it further into the back of their mouth by using that second pair of jaws.
Ooh.
So they're called like pharyngeal jaws. They are very cool looking. They're scary looking if you were to look down the mouth of an eel, but they are very cool.
That makes me think of the movie Alien.
Yes.
Had that little
It is
jaw
something
within the mouth.
very similar to that.
Who are also known as xenomorphs.
Xenomorphs.
Aliens are xenomorphs?
In that movie, right?
Yep.
Yeah.
So eels of the order Anguilliformes lack a pelvic fin. So you will see a longer, hold on.
So like a longer anal fin, or the anal fin and dorsal fin just kind of merge all together with the caudal fin?
Well, they do that.
Okay.
Ah, look at how different the cliff face is on the left-hand side and the sort of middle view.
Looks like it sheared off, or it got more
Yeah.
slumping happened.
Yeah, and it looks recent, at least in the way of, okay, hypothesizing. It looks recent because I don't see fauna on it, and also there isn't a layer of sediment coating the surface. It's a very cleanly sheared and flat-looking faces to it. So yeah, that seems like a recent slump.
That's cool looking. We have a question of, has anyone ever seen a rock fall or landslide underwater?
Yes.
Yes.
I've only heard about it, so I don't know all of the details, but I know that Charlie Paul at the Monterey Bay Aquarium Research Institute has been studying submarine landslides. This is a common thing. They're called turbidity flows. And so they wanted to understand how often turbidity flows occur. They knew that there are these big landslides that have a sort of a head at the front and then a big plume or cloud of sediment behind. And so they were putting out sensors to do an ROV dive.
You're just going to get tugged here.
And then a turbidity flow happened. And so the ROV experienced
It's going to try and...
Oh, that
being
had to be
inside of a
exciting.
turbidity flow. Oh, yes. There were a lot of white knuckles, but also, you know, after the fact, when the ROV was okay, they were really thrilled that they were able to see that happen. And now they have sensors out that are about the same buoyancy as sediment, and so they've set them out in the canyon, and if there's a landslide that happens, they'll tumble along with the landslide and collect data, like how fast the landslide was and where the sediment ends up. So yeah, they've been studying that for a while.
That's pretty cool. I don't think I would like to be stuck in an underwater landslide. Would a resulting tsunami affect you after a underwater landslide? I feel like it would kind of create an underwater wave. I'm not sure if it would really be a tsunami. I guess it would depend on the size of the sort of slump.
I don't know the answer to that.
I don't either.
Yeah.
I'm postulating.
My theory on that would be that an underwater landslide doesn't change the volume of water enough to create a tsunami. When an earthquake happens and the actual floor shifts, then a large quantity of water can either be pushed up or fall down, and then that rapid change of a large volume of water creates the tsunami when it reaches when the waves propagate to the continental shelf.
Yeah, I would agree with that. So I think this will be a disturbance sort of underwater and sort of forces flowing one way or another, but as into a tsunami, it wouldn't quite be that.
A landslide is just existing mass moving around on the surface of the bottom. It's not changing the volume or the space that the water occupies.
Yeah.
So to that person who asked the question of, has anyone ever been in a turbidity flow, Mbari actually wrote a write-up about it. There's a publication in a research journal, and then there's also an article written by Mbari as a press release called Swept Away, Deep Sea Robot Caught in Underwater Avalanche Yields New Scientific Insights. And they've got a visualization. They have a YouTube video that shows it, so you can actually see what one of those underwater landslides looks like and hear their description of it.
Oh, that's pretty neat. I'm definitely going to check
Old
that
trumpet
out.
sponge.
Is it a trumpet? Oh, dead trumpet sponge. Yes, on the left. I was looking at the white one. That looks like umbellula. Doesn't it?
I think you're right.
That's the first umbellula we've seen.
Yeah.
Neat. On our watch, at least.
That's a sea pen.
What is a sea pen? Tell me more.
Sea pens are related to corals.
Okay.
So they're cnidarians,
They
Sure.
are.
which means that they have stinging cells with nematocysts inside, because remember, that's a
275
unifying
meters.
feature of cnidarians as a group.
Ah,
And sea pens live on the seafloor and have
60
an
meters.
anchoring basal peduncle, which projects into the seafloor and kind of keeps it from blowing away.
peduncle?
A peduncle.
Peduncle. That is one of my favorite words.
Foot,
Peduncle.
foot something.
It's a good one.
And
It's just
they are octocorals.
Ooh.
So if you look real close, you'll have 8 polyps, or 8
Eight
tentacles for each polyp.
Mhm. Oh, these are neat sponges. Hey, it's that one.
Is that
That's
Is that a coral?
octocoral.
That's the one we've been seeing look like an aloe plant.
Yep.
Yes. We had a name. We thought that was yesterday. Who was it? Bathyalcon?
Is that what that is?
Getting there. Oh, lots of crinoids here. More sea anemones. Anywhere they can be not covered in sediment, it looks like they're sticking right on the edge of that cliff face.
You're
There's another
rapidly
mushroom coral.
approaching.
That's a beautiful view of a mushroom
You're
coral
rapidly
right there.
approaching that cliff. Terrain. Pull up.
So it's Bathyalcyon robustum was that aloe-looking soft coral. Maybe we'll see another.
I think these features are really interesting because we can see such a diversity of life, and they're all using the edge of this cliff to try to get an advantage of collecting more food.
Yeah, I agree. There are definitely certain areas where... oh, there's another predatory tunicate.
Hi, tunicate.
It's that guy. Certain areas where particular groups that have particular feeding strategies or anchoring styles tend to do better. So this is one where animals that capture food from the
And
water
yet-
really benefit.
It's true. It's really interesting to see. Is that an anemone that's all closed up?
It is.
Yeah.
Wake up, anemone.
Good
It's
catch.
morning time.
Ooh, this looks fresh.
Oh, it does.
Wow.
Yeah, it looks like it's an overhang, and maybe it's slumped off. Doesn't have that fine layer of sediment on it.
Mhm.
So maybe we're looking at the clay, perhaps.
Oh. Wow.
I would love to go down and take a look, but we have to keep moving.
No, that's fine. Thank you.
No worries. It's great. Just
We
to
want to
see
keep
this.
moving.
I have no choice at this point. Getting towed.
It works for us.
Mhm.